Hola a todos, my name is Iván Carrera and this is my work, Machine Learning Models for Predicting Disease-Related Biological Interactions, in the Doctoral Consortium on Biomedical Engineering Systems and Technologies, BIOSTECH. Let me introduce myself. I'm a professor of the Department of Informatics and Computer Science of the Escuela Politécnica Nacional in Ecuador. My research focus is on big data and machine learning for bioinformatics. I'm also a writer and science communicator at papacientifico.com. I'm a student of the doctoral program on computer science of University of Porto in Portugal, which is a research-oriented program with a duration of 3 years. I'm currently in my 3rd year. My advisor is Professor Inés Dutra. I work in cooperation with the Chemo Bioinformatics Research Group of Universidad de las Américas in Ecuador. My co-advisor is Professor Eduardo Tejera. Now, my research problem. In silico drug repositioning is an essential part in the drug discovery process. Drug repositioning is the process of finding new uses for existing drugs. An effective approach for drug repositioning is the analysis of biological interactions. These interactions can include drug-disease interaction and drug-cell line interactions. Drug repositioning works by testing biological interactions or effects of drugs on genes, protein binding, and molecular pathways affecting the phenotype and genotype in humans. In general, for discovering new biological interactions, we need to represent the knowledge of biological entities such as drug, diseases, cellular lines, proteins, and so on. Current methods for drug repositioning on cellular lines either need gene expression or are valid only for a small set of cellular lines. Representing cellular lines by gene expression is frequent and is very used, but it is impractical for a general method. To be able to represent cellular lines by their gene expression, we will need to have the genes that are triggered by a drug. So, if we would try to do that for every drug-cell line combination, that would be impossible. And other methods for drug repositioning for cellular lines are only valid for a small set of cellular lines, making that there are no general methods for cell line representation. So, we identify a lack of a general and comprehensive model for discovering drug-cell line interaction. In another domain, disease representation is a complex issue. Disease representation methods include networks, pharmacokinetic and pharmacodynamic models, hierarchical modeling, and agent-based modeling. The current methods for gene-based disease representation deliver results that differ very much. There are a large set of methods for disease representation based on gene expression. We would need a method for summarizing and equalizing results, having these results from every disease representation method. Finally, once diseases and cellular lines are represented, we will need to discover drug interactions with such representations. The goals of our work are divided as a set of 4 computational methods. The first is a method for representing cellular lines. The second is a method for discovering new drug-cell line interactions. These two have already been accomplished. Third, a method for representing diseases. This is based on gene expression. And fourth, the discovery of new drug-disease interactions. The stage of our research is this. What has been accomplished are these 4 works. First, cell line representation, which is a method for the computational representation of cellular lines based on text mining. In that work, we process the scientific literature on cell lines, and using support vector domain description, we have been able to represent and differentiate a set of 200 plus cellular lines. In this work, we do not use gene expression information, and we try to make this method as broad as possible. We can broaden this method, and this has a compromise with the precision of the representation. Okay. Second, for the cell-line drug interactions, we have two works. First, cell fishing, which is a method for predicting cell line sensitivity to chemical compounds. Cell fishing predicts the activity of chemical compounds on a set of cellular lines, 750 cellular lines. And this is based on the chemical similarity of compounds with reported activities. In this case, the input for our work can be a novel drug or a novel chemical compound, and with computing the similarity of this input with a set of trained chemical compounds, we can predict which cellular lines can be the target of this chemical compound. And last, the cell line drug interaction discovery, which is a work presented here at Health Informatics, is a method for recommending drug-cell line interactions, and this is based on a recommender system. This recommender system compares pharmacological affinity profiles of cellular lines and calculates a probability of affinity with unknown or unreported compound interactions. So we can predict how is every drug in the set is going to interact with every cellular line in the set. Okay, so this is a very interesting work, and can be combined with the cell line representation. What is to be accomplished, and we hope to finish these two stages by the end of the curricular year, is disease representation, which is a method for representing diseases based on gene expression and drug sensitivity. This work has already been deployed and developed, but we lack a few experimental results, and mainly the research paper. And lastly, the disease-drug interaction discovery, which is a method for discovering drug-disease interactions. This is a current work at University of Porto, and we are working with a set of 20 diseases and the corresponding over 100 phenotypes of this disease. Okay, so the results of my work are this published work, and we hope to finish this and have an outcome of a methodology that can represent and can predict the interactions of cells and diseases with drugs, and mainly to have the discovery of new applications of known drugs on diseases and cellular lines. So, thank you for your attention, and now I listen to your questions. Thank you.